Hello students, I am teacher Svetlana and in our today's lecture we will learn about cell, kinds of cell, that is a prokaryotic cell and a eukaryotic cell, which is covered in the chapter Cell Structure and Organization. So let's begin. Cell. Cell is called a structural and functional unit of life of all living organisms, capable of independent existence and can perform all functions of life. To see cells clearly, we need a microscope. Large cells can be seen through simple microscope, but to see smaller cells, we require a compound microscope. Simple microscope can magnify images 50 to 100 times, but a compound microscope can do so 1000 times or more. In the microscope we use in laboratories, a beam of light is used to make things visible. Hence it is called as a light microscope. To see interior of a cell, we need electron microscope. It can magnify image up to 5 lakh times. So, with the help of the microscope, we can study a cell. There is no typical shape of a cell. Cells may be spherical, rectangular, flattened, polygonal, oval, triangular, conical, columnar, etc. Cell size varies greatly in various plants and animals. Some of them are not visible to the naked eyes. Some are barely visible, while some are microscopic. The smallest cell size can be seen in mycoplasma, that is around 0.3 micrometer in length. Bacterial cell size is 3 to 5 micrometers, while the largest size of a cell is seen in the ostrich egg, nearly 15 centimeters. Longest cells are the nerve cells. Here is an image of a typical cell structure. Cell theory and totipotency. Students, you already know that cell theory was proposed by Schwann and Slidin. However, in this theory, there was no explanation about the formation of new cells. It was Rudolf Virchow in 1855 who explained for the first time that new cells are formed by cell division from the pre-existent cells. This theory was called as omnis cellula acellula. In later years, advanced research in cytology is the study of cell, led to modifications in cell theory, which is now known as modern cell theory. Then students, we need to know a phenomenon called as totipotency. It is the capacity or the potential of living nucleated cell to differentiate and divide to form any other type of cell and thereby a complete new organism. A cell is totipotent because it has the entire genetic information of the organism in its nucleus. Embryonic animal cells are totipotent and termed as stem cells. Stem cells have great medical application, including cure for diseases. In other words, stem cells have a lot of potential in finding a cure for genetic disorders. Have a look at this image here. This here are called as the embryonic stem cells. So this is the zygote which is formed after the fertilization, that is the fusion of the male and female gametes. So these cells are called as embryonic stem cells. They get divided into the ectoderm, mesoderm and endoderm, and further they give rise to different types of cells, and furthermore this contributes in giving rise to a new organism. So this phenomenon is called as totipotency, the capacity or the potential of the nucleated cells to divide, differentiate and give rise to a complete new organism. Kinds of cells. Living organisms are grouped into two main categories, the prokaryotes and the eukaryotes. The prokaryotes have simple cellular organization, while the eukaryotes exhibit high degree of organization. Let us have a look at each of these types of cells. The first one is the prokaryotic cell. The cell in prokaryotes show following main features. It has chemically complex protective cell envelope, However, it does not have well-defined nucleus and other membrane-bound cell organelles. Here is an image of a prokaryotic cell. You can see that it has a complex protective outcoat, but it does not have a well-defined nucleus. The nuclear material is just suspended inside the cell. Cell envelope is a three-layered structure with outer glycocalyx, middle cell wall, and inner plasma membrane. So you can see these three layers. Glycocalyx is present as either a slime layer or a loose sheet or a capsule, which is a tough sheet. Bacteria are better observed when they are stained. Further, when we are trying to understand the prokaryotic cell, let us look at the characteristics ahead. The most followed staining method is gram staining, which was developed by Danish biologist Hans Christian Gram. So gram staining is a method used for staining the bacteria. It helps to identify the type of bacteria we have, and also it helps us to study the structure of the bacteria. The cell wall is made up of peptidoglycan in gram-positive bacteria and murines in gram-negative bacteria. It gives mechanical strength to the cells. We have two types of bacteria which are identified with the help of the gram staining, and they are called as gram-positive and gram-negative bacteria. Cell membrane is a phospholipid bilayer. All these structures give protection to the cell and also help in intracellular transport. In motile bacteria, either cilia or flagella are found. Both are driven by rotary movements produced by the basal body, which works as motor. Other parts are filament and hook. So this here is the flagella, which is used for the process of locomotion. Some other surface projections, such as tubular pilia, which help in intracellular communication, and fimbriae for clinging to support. So they also have such additional projections present on their body. The cell membrane shows infoldings called as mesosomes, which help in cell wall formation and DNA replication. Some bacteria, especially photosynthetic cyanobacteria, show more longer extensions called as chromatophores. Now these chromatophores carry photosynthetic pigments. The cytoplasm contains dense particles called ribosomes, which help in protein synthesis. So in the cytoplasm, one important organelle is the ribosomes, which helps in the synthesis of proteins. Ribosomes are described by their sedimentation rates in Swedberg units. Bacterial ribosomes are 70S, composing of large subunit 50S plus a small subunit 30S. Sedimentation rates, students, means when these organelles are being separated out for the purpose of studying, they are subjected to a process called as centrifugation, which is a separation technique. Now here they separate out from each other depending on their molecular weight differences. So at that time their sedimentation rate is termed as Swedberg unit. Eukaryotic cells. This is the second type of cells in which the nucleus has a definite nuclear membrane are known as eukaryotic cells. These cells exhibit presence of membrane-bound organelles. For example, we have cells of protists, plants, animals, and fungi. The eukaryotic cells have different shape, size, and physiology, but all the cells are typically composed of plasma membrane, cytoplasm, and its organelles, which are mitochondria, endoplasmic reticulum, ribosome, Golgi complex, etc., and a true nucleus. So if you have a look at this image here, you can see a well-defined nucleus is present. Then you have the cytoplasm, and you have these tiny organelles which are membrane bound. So you are separated out from the cytoplasm. So you can identify them as individual small organelles. This is the mitochondria. Here you have the lysosomes, Golgi bodies, ribosomes. This is the cytoskeleton. Each This is the endoplasmic reticulum. Each of these organelles we will see in detail in the chapter ahead. Your students, for your quick reference, I have put up a difference between the prokaryotic and the eukaryotic cell. You can pause the video here for a while and have a look at the differences. This here, student, is the image of a typical plant cell and animal cells. You must have seen these images in your previous classes. But these images are put here for just a quick recap so that you will be able to understand the difference as well as the different types of organelles that are present in each of these cells. Here, students, I have also put up a difference between the typical plant cell and the animal cells. Most of you must be aware of these differences, but this is just a quick recap. So it will help you to understand the organelles that are present in each of these cells. Thank you, students, for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe, and keep learning.